Hello, let's understand the basic steps of recombinant DNA technology. Suppose you want to produce insulin on large scale, and insulin is produced by a pancreas in human beings. So then we isolate a fragment from the pancreas which contains insulin gene, which you can see here. This is the fragment, and then we require a host, which is E. coli. E. coli is a bacteria, and as you can see, it has circular DNA, which is also called plasmid. So I have shown you here just 4 plasmids, but actually the bacteria contains from 3 to 600 plasmids. So these plasmids confer special properties to the bacteria like antibiotic resistance and maybe ability to degrade hydrocarbons. So we isolate this plasmid, as you can see here, and then this is just a presentation of the plasmid. It's shown in enlarged plasmid. So this plasmid is isolated and it's cut with enzyme, which are also known as molecular scissors. These are restriction enzymes, and you can see the examples like EcoR1 and HIN3. So why do we use these enzymes, identical enzymes, for cutting our plasmid and cutting our insert, which is the gene of interest, with the identical enzyme, so as to create identical ends. So you can see the staggered ends are created and it's easy to join them. So here you have the recombinant plasmid, but how do we join this insulin gene with the plasmid? It is with the help of a glue, which is the molecular glue, and it is enzyme ligase. Now this is called a recombinant molecule or recombinant plasmid. Now this recombinant plasmid is put back into the bacteria, which you can see here. Okay, so there's a possibility that some bacteria might not take up this recombinant plasmid. So we need to differentiate between the bacteria, the ones which have insulin gene and the ones which do not have. So we have certain selection methods like antibiotic resistance or blue-white colony selection methods. So we just select those cells and we allow them to propagate. And so we have a number of bacterial cells, recombinant bacterial cells, which produce insulin on large scale. So just to recap, what are the 4 basic steps? First is isolation of DNA fragment with the gene of interest from pancreas, if we are considering insulin. And the second step would be generation of recombinant DNA molecule. The third step would be transformation, that is introduction of rDNA into the host cell. And the fourth one would be selection of host cells with rDNA by using certain selection methods. I hope these points are clear to you. Otherwise, I am always there with you. Thank you.